Aquaporin is a transmembrane protein that is synthesized from various processes such as transcription and translation, and will then be modified in the rough endoplasmic reticulum Golgi apparatus to be embedded in the phospholipid bilayer for its function. The first process for transcription will be initiation. RNA polymerase binds to the promoter region consisting of a large amount of adenine and thymine bases. These adenine and thymine bases are easier to break due to the double hydrogen bonding. This separates the two strands of DNA with one template and a non-template strand. Moving on, the second phase of transcription will be elongation. RNA polymerase moves along template strand from 3' to 5' direction, unwinding the DNA helix. 3 ribonucleotides join with the strand through complementary base pairing through hydrogen bonding. The adjacent ribonucleotides are then joined via phosphodiester bonds. The new transcript is synthesized from 5' to 3'. The last process of transcription will be termination. The elongation of pre-mRNA occurs till the transcription stop site is reached. Then it releases the newly formed mRNA and the DNA rewind. Now we move on to the post-transcription modification. Addition of 5' capping with 7-methylguanosine serves as the recognition signal for the small ribosomal subunit to assemble and begin translation and facilitates the export of mature mRNA from the nucleus. Poly-A-tail helps to protect degradation of the mRNA by ribonucleases, making it a more stable template for translation and directs the export of mRNA through nuclear pores into the cytoplasm. Now we move on to RNA splicing, which requires ATP. Spliceosomes remove introns and joins the exon together. Now this makes it a more stable template for translation and directs the export of mRNA through nuclear pores into the cytoplasm. Now the following is how a spliceosome splice the exon together and remove the introns.
This process is called amino acid activation. This process requires amino acyl tRNA synthetase to go and bind with a tRNA which is complementary to the amino acyl tRNA synthetase with a complementary amino acid. This is how it will look like. This process requires ATP, adenosine triphosphate, which will then bind with the amino acyl tRNA synthetase and then break a single phosphate bond. This process will release a large amount of energy that will allow this process to happen. The resultant product will be amino acyl tRNA. For the translation initiation complex to be formed, the small ribosomal subunit has to bind with 7-methylguanosine and begin the translation initiation with the large ribosomal subunit and the tRNA molecules holding methionine. This will be then called the translation initiation complex. The whole mRNA strand consists of a 5' cap, which consists of 7-methylguanosine, and a poly-A-tail containing a lot of adenine molecules. The translation will begin when the tRNA molecule from the ribosomal subunit in the amino acyl site will move to the peptidyl site, transferring its amino acid over to the amino acyl site and then to the exit site. It will be shown in our pictorial representation over here. So it moves to AUG, which codes for methionine, and then it continues moving to the second codon, transferring tRNA, the first tRNA to the P site. Another tRNA joins, complementary to its mRNA. The amino acid from the first tRNA molecule will then be transferred over to the other tRNA molecule with the help of peptidyltransferase. This breaks an ester bond in the first tRNA molecule, followed by joining the two amino acids with a peptide bond. It will then move again to the next, move again to the next codon, and another tRNA molecule will come in. At the exit site, the tRNA molecule leaves into the cytosol of the cell. The whole process repeats until it reaches a stop codon, which can be coded for including UAG, UAA, and UGA. A release factor will allow the polypeptide chain of the ribosome into the RERs, which is the rough endoplasmic reticulum, for further modification into a functional protein such as aquaporin.
Next, we'll be moving on to talk about the movement and modification of aquaporins. The vesicles containing aquaporins pinches off from the rough endoplasmic reticulum and move towards the cis phase of the Golgi apparatus and fuses with the Golgi apparatus. The Golgi apparatus then sort, packages, and modify the aquaporin during the transit from the cis region to the trans region of the Golgi. After aquaporin is being modified, the trans phase of the Golgi apparatus gives rise to vesicles which pinch off and travel to the cell membrane. Next, we'll be moving on to talk about the bonding of aquaporin on the cell surface membrane. When the vesicle carrying the aquaporin reaches the cell surface membrane, the vesicle is integrated into the cell surface membrane.
Upon translation, the signal peptide is recognized and bounded by a protein-RNA complex called signal recognition protein, also known as SRP. SRP acts as an adapter that brings the complex to the SRP receptor on the rough endoplasmic reticulum. Then the SRP leaves. SRP is part of a multi-protein translocational complex which contains a protein pore and a signal cleaving enzyme. Translation resumed. Growing polypeptide chain move through the protein pore into the lumen. Signal cleaving enzyme remove the signal peptide. Upon termination of translation, endomembrane polypeptide will remain partially embedded in the endoplasmic reticulum. Polypeptides that are secreted will be released fully into the rough endoplasmic reticulum. RER is the site associated with protein synthesis, modification, and transport. New polypeptides produced by fixed ribosomes are released into the cysteinate of the rough endoplasmic reticulum. Within the cysteinate space, the polypeptides start folding en route to form functional proteins. Proteins may be chemically modified by the attachment of carbohydrates forming glycoproteins. Most of the proteins and glycoproteins produced by the rough endoplasmic reticulum are packaged into a small membranous sac that pinches off the tip of the cysteinate. These membranous sacs, which are pinched off, are called transport vesicles.